Hey class, welcome back. This week we are in week 12 of our 15-week course. Uh, After this week we have 2 more lectures. So today we are diving into the spinal cord, spinal nerves, and spinal reflexes. Our spinal cord and spinal nerves contain circuits of neurons that mediate our fastest responses to environmental changes, and those are called our reflexes. So when something happens in the environment, you can react extremely quickly, and those reflexes are mediated at the spinal cord itself. It is also the site for integration or summation of excitatory postsynaptic potentials and inhibitory postsynaptic potentials that arise either locally within the cord or are triggered by impulses received from the peripheral nervous system. It also provides a pathway for impulses traveling to and from the brain. So sensory nerve impulses are traveling up the spinal cord toward the brain. Motor commands are traveling from the brain down the spinal cord and then out to the peripheral nervous system. Reflexes are rapid automatic nerve responses triggered by a specific stimuli. We're going to talk about a few different reflexes. Spinal reflexes are controlled by the spinal cord, not by the brain. So information will eventually be sent to your brain so that you can process what happened. But if you put your hand on a hot stove, you're going to pull it away from that stove before you're ever aware that it actually is hurting. And that is because the reflex is mitigated at the spinal cord level. Our gross anatomy of the spinal cord, it is about 18 inches long and a half an inch wide. It ends between the levels of L1 and L2. Most people think your spinal cord travels all the way down your spine, but it actually ends between L1 and L2. It is symmetrical bilaterally, so it has grooves that divide it into a left half and a right half, and there's an anterior groove and a posterior groove. On the posterior side, it's called the posterior median sulcus. And on the anterior side, it's the anterior median fissure. And if you remember, a fissure is deeper than a sulcus, so the anterior median fissure is a deeper groove on the anterior surface, whereas that posterior median sulcus is a more shallow groove on the posterior surface. We'll see some pictures in a few slides. The amount of gray matter is greatest in the segments of the spinal cord that are dedicated to sensory and motor control of the limbs. The gray matter is our neuron cell bodies, and so where we need more information to be processed and transmitted, we're going to have additional gray matter. So we have expansions in those areas called the cervical enlargement and the lumbar enlargement. The cervical enlargement spans from vertebrae the level of C4 down to T1, so that's at the base of your neck. And the nerves that exit from between C4 and T1 are going to supply your shoulders and your upper extremities. The lumbar enlargement is from T9 to T12, and nerves exiting that region are going to supply your pelvis and your lower extremities. And do notice that the lumbar enlargement is in the thoracic region. So T9 to T12 is your lumbar enlargement. The cervical enlargement, the nerves that leave the cervical enlargement create what we call the brachial plexus. A plexus is where a group of nerves come together and sort of intermingle and then move on to supply other regions. So the brachial plexus, it arises from the cervical enlargement, and the lumbar or the lumbosacral plexus arrives from the lumbar enlargement. So here's a picture You can see up on the top here, this is the cervical enlargement. So you have a fair amount of gray matter in the middle, shaped kind of like a butterfly, with white matter surrounding it. And then in the thoracic region, there's a lot less gray matter because we don't have all of those nerves exiting to supply the arms or the legs. Once you get down into the lumbar region, you end up with much more gray matter as you have all of those nerves exiting that are going to go and supply your pelvis and your lower extremity. You can also see here, if I delete that, this is our anterior median fissure, and then this is our posterior median sulcus. The distal end, or the end further away from the head, where it ends between L1 and L2, ends at a thin tapering called the conus medullaris. We see that right here, conus medullaris. So that is that distal tip of the spinal cord where it ends, and that is just below the lumbar enlargement. That tip is then tethered all the way down into the sacrum by the filum terminale, which is a thread of fibrous tissue that goes from the tip of the conus medullaris down into the coccygeal ligament. And so it's holding the spinal cord in its position and keeping the spinal cord from bunching up and moving superiorly. Then we also have the cauda equina, which is all of the nerve roots that exit from the spinal cord above the conus medullaris. They come down in cauda equina. Cauda is like a tail, so cauda equina means horse's tail. So it's this large group of spinal nerves that are kind of bound together in the spinal canal below the conus medullaris. That is our cauda equina. When we're looking at individual segments of the spinal cord, we have nerve roots that exit at each level, and each segment has a dorsal root ganglia on either side. So here's our dorsal root ganglia. We have two of them there. And what those are a collection of neuron cell bodies of our sensory nerves. So our sensory nerves have their dendrites out in the periphery. And then, so here we can draw dendrites out in the periphery. And then the nerve comes up like this. And then the cell body is in the dorsal root ganglia. And then the axon leaves the dorsal root ganglia and comes into the posterior horn, which is on the back side of the spinal cord, and it synapses in the posterior horn. So if you remember, our sensory nerves are unipolar nerves. So they have the dendrite, and then the cell body is kind of off to the side, and then the axon comes like that. So it has like one process with a single dendrite and a single axon. And so the cell bodies of those unipolar nerves that are our spinal nerves are collected in the dorsal root ganglia. And so then from the dorsal root ganglia to the posterior horn is the dorsal root of the nerve. So each spinal nerve, so the spinal nerves they form just outside or inside the intervertebral foramen. So if you remember back to what our spine looks like, we have vertebral bodies stacked up upon one another. And then just posterior to the vertebral bodies, when you have two vertebrae together, there is a space in between the two vertebrae called the intervertebral foramen. And the spinal nerve is going to form in the intervertebral foramen. So once the spinal nerve forms from the dorsal root and the ventral root. So there are two roots that leave the spinal cord. One of them is on the posterior side, the dorsal root, and one of them is on the anterior side, the ventral root. So the dorsal root is going to be all sensory information because it's the neuron cell bodies and axons of sensory nerves. The ventral root is motor information. And so when the ventral root brings its motor information and the dorsal root brings its sensory information, the actual spinal nerve itself becomes a mixed nerve, which means that spinal nerves are carrying both motor information and sensory information. Sensory information is afferent, meaning it's being collected in the periphery and brought toward the central nervous system. Motor information is efferent, meaning it's being issued in the central nervous system and then extending, traveling outward to the peripheral nervous system. So if we were to take the spinal cord and cut it in half at the midline, the posterior portion is all sensory and the anterior portion is motor. I also wanted to mention that there is no ventral root ganglia because our motor neurons are multipolar neurons, so they do not have a collection of cell bodies. The cell body is right around the dendrites, and then we have the long axon, which goes to the next cell body, and so there is no ventral root ganglion. There is only a dorsal root ganglion, and that is because our sensory nerves are unipolar. Okay, so our spinal cord is extremely important, and when things are extremely important, we've learned that they are protected heavily. So we protect our spinal cord in three ways. They are protected by our meninges, which are connective tissue coverings of the spinal cord. They are enclosed by the spinal canal in the bony vertebrae, and they are surrounded by our cerebrospinal fluid, which is cushioning and nourishing to the spinal cord. So we'll start by taking a look at the meninges. The meninges are specialized membranes that isolate the spinal cord from its surroundings. The brain and the spinal cord have a very sensitive homeostatic state at which they live, and they need to be isolated from all of the surrounding tissues. So the meninges help in that isolation. The functions of the meninges are to protect the spinal cord, to provide a place for our blood supply to move and bring blood into the spinal cord, and they become continuous with the cranial meninges. So the beauty is we're going to learn spinal meninges this week, and next week when we get into the brain, we'll learn cranial meninges, and they are the exact same 3 layers as the spinal meninges. Meningitis is a viral or bacterial infection of the meninges. It can be very painful and very dangerous. Viral infections are usually self-limiting. Bacterial infections can cause death if they go untreated, but you get this severe inflammation in the meninges due to that infection, and then that inflammation presses on your spinal cord and can cause all kinds of interruption of normal nerve functioning. So our meninges are connective tissue coverings that encircle the spinal cord and the brain, and they are made up of 3 layers: the dura mater, the arachnoid mater, and the pia mater. The dura mater translates as tough mother, and it is tough. It is the fibrous outer layer. Cranially, it fuses with the periosteum of the occipital bone, so it is held really tightly in place with the occipital bone, and it becomes continuous with the dura mater of the cranium. Caudally, so towards the tail, it tapers into a dense cord of collagen fibers and joins the filum terminale in to attach into the coccygeal ligament. So the dura mater and the filum terminale are going to keep the spinal cord from moving superiorly and inferiorly within the spinal canal. The epidural space is the space between the dura mater and the walls of the vertebrae. So if we're looking right here, you can see that the dura mater is shown in blue, the vertebrae is shown in tan, and there's that very small space between the vertebrae and the dura mater, and that is called the epidural space. And it is filled with a loose connective tissue and adipose tissue for cushioning. And this is the site of epidural injections. So if you go and you have an epidural injection, they inject between the bone and the dura mater and to anesthetize spinal nerves. The arachnoid matter is the middle layer of the meninges. It is a simple squamous epithelial membrane that surrounds the arachnoid matter. And then the arachnoid matter looks kind of like our trabecular bone. When you think about that like web-like formation of trabecular bone, that's what the arachnoid matter looks like. It looks kind of like a spider web. So it's this thin yet web-like meningeal layer. The arachnoid matter is made of collagen and elastic fibers. The subdural space is the space between the dura mater and the arachnoid mater. So epidural is outside of the dura, subdural is under the dura. So the subdural space is between the dura mater and the arachnoid mater. The subarachnoid space is the space between the arachnoid matter and the pia mater. So this contains collagen elastic network and the arachnoid trabeculae, and the arachnoid space is the subarachnoid space is filled with our cerebrospinal fluid. So we have cerebrospinal fluid that is always floating all around the spinal cord, and it is within the subarachnoid space. Cerebrospinal fluid carries dissolved gases, nutrients, and waste products. And a spinal tap is where you go in and you actually withdraw cerebrospinal fluid from the subarachnoid space, and you can analyze it to see, like if you suspect that somebody has meningitis, those viruses or bacteria would show in the cerebrospinal fluid. So if you were to withdraw some and look at it under a microscope, you could identify the viruses or bacteria that are infecting the area. The pia mater translates as delicate mother. It is the innermost meningeal layer, which is a mesh of collagen and elastic fibers that is actually bound to the spinal cord itself. The spinal cord has denticulate ligaments, which are paired. They're shown here. So there are these little areas of connection where the pia mater is connecting to the dura mater. And what this does is it stabilizes the spinal cord so it doesn't move side to side. So the dura mater and the filum terminale are keeping the spinal cord from moving superiorly and inferiorly, and the denticulate ligaments are keeping the spinal cord from moving laterally. Our blood vessels run along the surface of the pia mater within the subarachnoid space. So you can see here in the center, coming up that anterior median fissure is a blood vessel. So we're going to look at the anatomy of the spinal cord itself. We talked a little bit about it, but we'll go more in depth right now. So the spinal cord is made up of gray matter and white matter. The gray matter is this butterfly-looking thing in the middle. The white matter is everything that surrounds it. So the white matter is that superficial layer, and it contains myelinated and unmyelinated axons of neurons. So myelinated and unmyelinated axons. gray matter surrounds this central canal. So here's our central canal, which is a hole right in the center. And the central canal is for the passage of cerebral spinal fluid. And our gray matter surrounds that. Gray matter is made up of neuron cell bodies, neuroglia, and unmyelinated axons. So the myelination of the axons is what gives the white matter its color. And then these cell bodies, which contain the nucleus and the nissl bodies and all of that stuff, is what gives the gray matter its color. And the gray matter has projections, which are called horns. So our gray horns are broken into posterior gray horns, anterior gray horns, and lateral gray horns. So what delineates anterior from posterior is about the level of the central canal. So all of this is posterior horn. So you have one there, and one here, and one there. And our posterior horns are sensory information. And we know this because our sensory nerves were coming up through the dorsal root ganglia and then synapsing in the posterior horns. So dorsal root ganglia, sensory, dorsal gray horn, sensory. And it's containing somatic sensory as well as visceral sensory. So visceral sensory is sensory from our viscera or our internal organs. So all of the sensory information from your body is in the posterior horns. The anterior horns, which are in this area, one here and one here, are motor information. And they are specifically somatic motor. Somatic motor. So the somatic nervous system is supplying your skeletal muscles. So the anterior gray horns are sending commands to your skeletal muscle, somatic motor. And it is shown here in that pink region. That is our somatic motor anterior gray horn. Last, we have the lateral horn, which we see over here. And the lateral horn is visceral motor. And visceral motor is motor information to the smooth muscles within your internal organ system. So visceral information is going to innervate smooth muscle. Somatic motor is going to innervate skeletal muscle. So our visceral motor is our lateral gray horns. Our somatic motor are the anterior gray horns. And all of our sensory comes into the posterior gray horns. We also have two regions called the commissures, and the commissures are where we have axons that cross from one side of the spinal cord to the other. We have an anterior gray commissure. I have to do this in small, which is here. So it's just that gray matter right in front of the central canal. We have a posterior gray commissure right behind the central canal. And then we have an anterior white commissure, which is in front of the gray matter, and it is surrounding the very inside of the anterior median fissure. In the peripheral nervous system, we've discussed the ideas of ganglia being a collection of neuron cell bodies. In the central nervous system, our grouping of neuron cell bodies are called nuclei. Sensory nuclei are dorsal, they're posterior, and they receive information from peripheral receptors. And motor nuclei are ventral or anterior, and they issue motor commands to peripheral effectors. So remember, a receptor is receiving information, and an effector is delivering a command. So this is the same information on this slide, but it just shows you that here's our posterior sensory information, receiving information from our peripheral receptors via the dorsal root ganglia. And here is our somatic motor and our visceral motor, and they would both send their commands out the ventral route, and those are motor commands. Our white matter is also organized in the spinal cord into columns. So our posterior white columns, which are here, are between the posterior median fissure and the posterior gray horn. So I just circled the right. It looks like the left, but remember we're in anatomical position. And so that would be the patient's right posterior horn, or posterior white column, sorry. And then the left posterior white column would be there. The anterior white columns are between the anterior gray horn and the anterior median fissure. So they cover this region. So there's one anterior white column, and then there would be another on the other side. And they also contain that anterior white commissure, where you have axons crossing from one side to the other within the white matter. And then our lateral columns are between the posterior horn and the anterior horn on either side. So those are our two lateral columns. Our last week, in week 14, we're going to talk some more about the tracks and that the organization of the white matter and how information is running up and down the spinal cord within specific areas of this white matter. So tracks are, that's what I said, in the white columns, they're bundles of axons that relay the same information in the same direction. So they are like pain and temperature. Pain and temperature run together and they run in the same direction. So they're going up toward the brain so that you can experience pain and experience temperature sensations. Ascending tracks are carrying information to the brain, so that's going to be sensory information. And descending tracks are carrying motor commands from the brain to the spinal cord and then out to the spinal nerves. So the summary of our spinal cord, our gray matter is covered by a thick layer of white matter. The gray matter contains sensory information in the dorsal portion and motor nuclei in the ventral portion. The white matter is made up of ascending and descending axons, and it's organized into columns. And columns are containing axons that send the same information in the same direction. And those axon bundles are very specific. So because the spinal cord is so highly organized, it's possible to predict the results of an injury to a specific area. So when I do this in class, I usually split you guys up into groups and I give you a little like study to consider. So what I usually do is say two athletes were both injured. Both of them are quadriplegic, but one can breathe on his own and one cannot. And the question would be why? And so you don't have all of that information yet, but with the information that you have, I want you to think about that for a minute, and then you can pause this while you think about it and then come back to it and I'll give you the answer. Okay, so our answer to why one athlete can still breathe on his own and one can't is because of the level of the injury at the spinal cord. So the ability to breathe on your own requires the contraction and relaxation of the diaphragm. The diaphragm is innervated by spinal nerves C3, C4, and C5. And when I was in school, we had a little song that said C3, 4, 5 keeps the diaphragm alive. And so the patient who can breathe on his own was injured below C5, so still can't move the arms or legs. So if you're injured at C6, you wouldn't be able to move anything below your neck. However, you would still be able to utilize your diaphragm and breathe on your own. If you're injured above C3, then you are quadriplegic and you would need to be on a ventilator because you would not be able to breathe on your own. Each of our spinal cord segments, like I talked about before, is connected to a pair of spinal nerves. The spinal nerves are made up by the dorsal root and the ventral root coming together in the intervertebral foramen. Every spinal nerve is surrounded by 3 layers of connective tissue, and it has connective tissue layers that support the structures and contain blood vessels. Peripheral nerves are interconnecting branches of spinal nerves made out of those plexuses that we discussed earlier. And peripheral nerves are also surrounded by these connective tissue sheaths. So we have 3 layers of connective tissue. And they are going to sound very similar to the layers of muscle tissue. So all you have to do is replace the word mesium from our muscle tissue with neurium for nerve tissue, and you will have it down pat with which one is which. So the epineurium is the outer layer outside of the nerve. It is a dense network of collagen fibers, and it's protecting the whole nerve. The perineurium is the middle layer, and it divides our nerves into fascicles. And fascicles are bundles of axons. So when you have a nerve, there are thousands, if not more, axons of individual neurons within that nerve. And so they are compartmentalized into fascicles, and the fascicles are surrounded by a perineurium. And then the endoneurium is the inner layer, which surrounds the individual axon itself. So here we have one neuron with a myelinated axon, so you can see the Schwann cells that we talked about last week. And then the endoneurium is this portion right here that would be covering over the Schwann cells and wrapping around that entire neuron or axon. This picture is on page 444 in your textbook. I would suggest that you take a look at it. So our spinal nerves are forming just lateral to the intervertebral foramen, where the dorsal root and the ventral root unite. Then they branch to form the pathways to their final destinations. So once we make it to our spinal nerve is going to branch and go in multiple different directions. And each direction is going to carry different information. So our first branch is the white ramus. And so the white ramus is going to carry visceral motor fibers, so those are autonomic fibers, to the sympathetic ganglia of the autonomic nervous system. So here's our sympathetic ganglia, and here's our white ramus. So the white ramus is carrying these blue fibers. You can see they start over here in the visceral motor in the lateral horn. They come out the ventral root and then down in what's considered the white ramus to this sympathetic ganglion. That is going to synapse on the unmyelinated nerves, which return from the sympathetic ganglion. And so those are going to form the gray ramus, which comes back up and splits to join the ventral ramus and the dorsal ramus. The dorsal ramus is going to go and innervate the back side of the body. So it contains somatic and visceral motor fibers, because this, you can see here, let me do this in green. Oh, that's not green, that's black. You get the idea though. That is the ventral horn, so that is a somatic motor nerve in red. And you can see that that comes through the ventral root and then splits into the dorsal ramus and then the ventral ramus. So the dorsal ramus is carrying, let's see, I'm going to erase all that. So the dorsal ramus is carrying both the somatic motor nerves, shown in red here, and the visceral motor nerves shown in black that have returned from this sympathetic ganglion. The visceral motor nerves are postganglionic fibers, and they're going to innervate smooth muscles and glandular tissues in the back side of the body. The somatic motor nerves are going to innervate the skeletal muscles in the back side of the body. And then we have our ventral ramus here, and that is also going to have the visceral motor neurons shown in black that are going to innervate the smooth muscle, glandular tissues in the body wall and the limbs. And then the somatic motor nerves shown in red, which are going to innervate the skeletal muscles of the body wall and the limbs. So you can see that this is where the nervous system starts to get complex, is where you have multiple different types of information leaving the spinal cord along the ventral root and then splitting and coming back together to form the dorsal ramus and the ventral ramus. So this slide just gives you the information. I presented it in the last slide, but it's showing you that the dorsal ramus contains somatic and visceral motor fibers that innervate the back. The ventral ramus is larger and it innervates the ventrolateral surfaces and the limbs, and it also contains somatic and visceral motor fibers. Our sensory nerves are really interesting. They are very specific to what part of the body is bringing information to what part of the spinal cord. So the dorsal, ventral, and white rami are also carrying sensory information because it's the motor information going out, but these nerves are mixed, so they're carrying back sensory information. But once it gets into that spinal nerve right at that intervertebral foramen, the sensory information splits off and goes to the dorsal root ganglia into the posterior horn, whereas the ventral root is bringing motor commands from the spinal cord out to the spinal nerve. So the spinal nerve is bringing sensory information back to the spinal cord through the dorsal root ganglia. So dermatomes are bilateral regions of the skin that are innervated by a specific pair of spinal nerves. So you can look at this picture and see this sort of map on the body. And we use this clinically to determine what spinal nerve might be compromised if we have a patient who comes in with, say, arm pain or neck pain or back pain, leg pain. So let's look at the arm here. So you can see with the arm so this is the front side of the body here, and this is the posterior side of the body. So C4 comes across the top of the shoulder. C5 is the outside of the arm. C6 is the lateral portion of the arm and into the thumb. C7 is going to be this posterior side of the arm and into the index finger. Sorry, this is getting ugly with my drawing. C8, and we do have 8 cervical spinal nerves, so there is a C8. C8 is this inside of your arm, so you can see the C8 here. And then T1 is the inside of your upper arm. So C8 is the inside of your forearm, T1 is the inside of your upper arm. And then you can do the same thing looking at the leg. So the leg is L1, L2, L3, L4 comes down the inside of the calf. L3 does. L4 crosses over the tibialis anterior muscle. And then L5, where's L5? They're not even showing L5 on here. But L5 comes across the front of your foot, and S1 is the back of your calf. And the feet are also split up for that L4, L5, and S1 you can determine from the feet. So this is a very important clinical tool that we use to determine if there's compromise at the spinal cord level itself, and if that is compromising the spinal nerve. And we test this using a pinwheel, which is just what it sounds, a wheel with the sharp little points all around it. And you run it up and down both sides along the same dermatome. And you ask, does this feel the same on both sides, or is it sharper on one side or more dull on the other side? And you ideally want them to feel the same on both sides. But if you have a nerve that's being compromised, you're often going to lose sensation on one side. This is shingles. So with shingles, you have an inflammation along an individual nerve root. So that you have this dermatomal pattern of, oops, sorry, I didn't mean to do that. But you have a rash that forms along that dermatome of that nerve root. And it can happen with any nerve in the body. But you do get that rash right along the root of the path of that nerve. It's not just a random rash anywhere. It travels right along the nerve that is irritated. Shingles is a sequela of a chickenpox, and it is treated with antivirals. So we talked about the spinal cord and we talked about the spinal nerves. So now we're going to look at peripheral nerves. And our peripheral nerves develop into nerve plexuses. So a nerve plexus is made up of the ventral rami of thoracic nerves T2 to T12. Or, I'm sorry, forget I just said that. The ventral rami of thoracic nerves T2 to T12 go directly to the body structures that they supply. They do not go into a plexus. The ventral rami of all other spinal nerves do form plexuses. So a plexus is a complex interwoven network of nerve fibers, and it is formed from blending the fibers of the ventral rami, maybe focus on that, ventral rami, of adjacent spinal nerves, and they control skeletal muscles of the neck and the limbs. We have four major plexuses that we're going to learn: the cervical plexus, the brachial plexus, the lumbar plexus, and the sacral plexus. I'm going to kind of skim over these pictures, but I'll show you that this is our cervical plexus, this is our brachial plexus, this is our lumbar plexus, and this is our sacral plexus. But we're going to look at these individually one at a time. So our cervical plexus includes the ventral rami of spinal nerves C1 to C5. I do want you to know for each of these what spinal levels are form the plexus and what the major nerve of each plexus is. And I'll tell you as we go which ones I want you to know, but I want you to be able to associate the names of the nerves with the plexus, but I'm not going to make you know the specifics of like what levels are contributing to each individual nerve. That's above our undergraduate A&P. So the cervical plexus innervates the neck. It also sends information to the thoracic cavity and the diaphragm. The major nerve of the cervical plexus is the phrenic nerve. The phrenic nerve is made up of C3, 4, and 5, and it controls the diaphragm. So these are the other nerves within the cervical plexus. The phrenic nerve is the major nerve, the one that I really want you to know. But the other ones, if you were to see them on an exam, I would love for you to know that ansa cervicalis is in the cervical plexus. Hopefully you can word associate cervicalis and cervical. The lesser occipital, transverse cervical, supraclavicular, and great auricular nerves are also part of the cervical plexus. And you can understand that if you look at the nerves. Occipital is going to be head, so that would be the highest plexus, which is the cervical plexus. Transverse cervical has the name cervical in it. Supraclavicular is above the clavicle, which would be cervical spine. And great auricular, auricular refers to the ear, so the great auricular would also be the cervical plexus. The brachial plexus is an extremely important plexus. It includes the ventral rami of spinal nerves C5 to T1. I would expect you to know that. And it innervates our pectoral girdle and all of our upper limb. Our brachial plexus is split into cords. We have a medial cord and a lateral cord and a posterior cord. We'll get a better look at that on the next slide. I do not expect you to know which nerve comes from which cord, but just know that there are 3 different cords. The major nerves of the brachial plexus are the musculocutaneous nerve, the median nerve, the ulnar nerve, the axillary nerve, and the radial nerve. Radial and ulnar, you should know, are in the arm. Axillary is the word for armpit. So you should know that is also in the arm. Everything in the arm is brachial plexus. So the 2 to memorize here would be musculocutaneous and axillary nerves. So here's our picture of how the brachial plexus is created. So we have the nerve roots here, the ventral rami of spinal nerves, C5 to T1. And they're going to come together, so you can see that C5 and C6 come together here. C7 has its own trunk, and C8 and C1 come together here. So this is going to form our inferior trunk, our middle trunk, and our superior trunk. So we have 3 trunks, and then those trunks are going to split and combine. So you can see that there's the superior trunk is going to send a contribution along with the contribution of the middle trunk and a contribution of the inferior trunk to the, what do we have here? This is the medial cord. Is that the medial cord? No, that's the medial cord. Yeah, so you're going to look at this to see exactly what contributes to what. But so this is our axillary nerve that leaves right here, right there. So the individual nerves are branches off of the cords, and the cords are created from branches off the trunks. So by the time you get down to, say, the axillary nerve, you have potentially you have pieces from C5, C6, C7, C8, and T1 that could be part of the axillary nerve. So I don't expect you to know which levels of the vertebrae are sending information to which nerve, but just know that there are 3 trunks, there are 3 cords, that the plexus is interwoven fibers from C5 to T1, and then know the names of the main nerves coming off of each plexus. I'll also point out here that your scalene muscles run from the transverse processes of your cervical spine down to the clavicle and the first rib. So they are kind of coming like this. And so you can see that the brachial plexus goes right through and around those scalene muscles. So when your scalenes are really tight and tense, they can press on the brachial plexus and cause pain, numbness, and tingling in the arm that could mimic a disc herniation. We'll talk a little bit more about disc herniation later, but you could see a disc herniation that was right here pressing on the C6 nerve root could cause pain down like any of those regions, but then a pressure from the scalene muscle on that same trunk here could mimic that pain. So we have orthopedic tests that allow us to determine the origin of pain more so than just the patient saying this is where it hurts. Here are our cutaneous distributions. So we talked about dermatomes and how the dermatomes are individual spinal nerve distribution. And then our peripheral nerves also have their own distribution. So whereas we had C6, C7, C8 here with our spinal nerves, our peripheral nerves are a little bit different. So if you have compression of a peripheral nerve, then the symptomatic portion of your nervous system, the skin that's going to supply, is going to be different. So you can see that ulnar nerve is the pinky and the medial half of the ring finger on both sides there. The radial nerve is mainly the back side of the hand, but it doesn't cover the posterior or the distal tips of those fingers. And the median nerve is going to cover the anterior palm, most of it, but it's going to split that ring finger right down the middle. Carpal tunnel, which is compression in the over the carpals, compresses the median nerve. So if somebody has carpal tunnel syndrome, then they should come in with their symptoms being in this yellow area. If their symptoms are over in this area, then carpal tunnel is not their issue because the ulnar nerve does not go through the carpal tunnel. I don't expect you guys to know all of that stuff, but I like to bring in some of the real world information to help you realize why we're learning what we're learning. So our brachial plexus, the main nerves that I want you to know are let's see, on this one, the long thoracic nerve is an important nerve. The pectoral nerves, you should be able to figure out the pectoral girdle. All of these things are in the region that the pectoral girdle is. And then these ones, I definitely want you to know all of those: radial nerve, musculocutaneous, median, and ulnar nerves. The lumbar plexus is made up of the ventral rami of spinal nerves T12 to L4. So T12 to L4 here. The major nerves are genitofemoral nerve, lateral femoral cutaneous nerve, and femoral nerve. And so I'd like you to be able to just recognize that those names are related to the lumbar plexus. And then the sacral plexus is a ventral rami of spinal nerves L4 to S4. And the major nerves of the sacral plexus are the pudendal nerve, which you'll learn a lot about, innervates the bladder and the reproductive system. And the sciatic nerve, which most people have heard of. People say, oh, I have sciatica, my sciatic nerve is killing me. And so the sciatic nerve is what innervates most of the leg. The sciatic nerve then branches into fibular nerve and tibial nerve. We often refer to the lumbar and sacral plexus together as the lumbosacral plexus because they do sort of come down and innervate similar regions. So you can see we're going to see a better picture of the sciatic nerve in a minute. This is showing us the foot distributions of these nerves, the sural nerve and the saphenous nerve and the tibial nerve. So you can see the tibial nerve does this whole bottom of the foot and the back of the ankle there. So from here you can see the sciatic nerve, which is a very large nerve. It is very large. It comes down to the knee and it splits into the tibial nerve and the common fibular nerve. And then we also have this little sural nerve, which is in the lower leg and supplies the foot. As you can see here, this is sural nerve, the very lateral side of the foot, and then fibular nerve is all of this yellow part of the dorsal aspect of your foot. So with the sacral plexus, again, I want you to just recognize which nerves are coming from the sacral plexus, the most important being the sciatic nerve and the pudendal nerve. So we've talked about our spinal cord, our spinal nerves, and our peripheral nerves, but something else that we got to consider is the actual neurons themselves. So our sensory neurons we have about 10 million sensory neurons, and they are collecting information from the periphery and delivering it to the central nervous system. In contrast, we only have about 500,000 motor neurons that are capable of delivering commands to the peripheral effectors. So we need to be able to sense everything, but we can utilize fewer motor neurons to issue commands because we're only going to issue so many commands. And then we have our interneurons, which we did talk about last week. Our interneurons are association neurons that help us to integrate information between sensory neurons and motor neurons. And we have 20 billion interneurons. So they're very important in interpreting, planning, coordinating signals that are incoming and outgoing. So our interneurons outnumber all other neurons by far. Neuronal pools are functional groups of interconnected neurons or interneurons that can stimulate or depress parts of the brain or the spinal cord. So they can have excitatory or inhibitory neurons that can either increase an impulse that's coming in or suppress an impulse that's coming in. Our neurons function in different patterns. So we're going to look at each of those patterns. So the first pattern is divergence here, where you start with a single neuron and then you split that information into 2 neurons, and then that splits into 3 neurons. So now all of a sudden with just 2 splits, we've gone from 1 neuron down to 6 neurons. And so it's a way of continually moving out and sending a greater and greater message. Convergence is the opposite. Convergence is where you have information from multiple neurons that are all converging onto one individual neuron. So that's gathering a lot of information and delivering it to one neuron. Serial processing is where you are taking information and just sending it along a chain from one neuron to the next, to the next, to the next. So there's no divergence and there's no convergence in serial processing. Parallel processing looks like divergence, but it's different because you take your one neuron and you split that information into 3 neurons, and then you undergo serial processing in those 3 neurons. So you can make a little bit more of a widespread information, but then that information is going to flow in one direction to a very specific region and not continue to spread out. And then our last one is our reverberation. And reverberation is a positive feedback mechanism. So what reverberation does is you have one neuron that's going to stimulate a second neuron, and that second neuron is going to stimulate a third neuron, but it's also going to have a branch that goes back and re-stimulates the first neuron. So when these are functioning, they're going to continue to stimulate the first neuron and continue to send the same signal. And these are going to function until they are inhibited by either some inhibitory stimulus, like an interneuron coming in and interrupting that signal, or by synaptic fatigue, where they've released all of their neurotransmitters and they have not been able to reassemble additional transmitters to continue to fire. Reflexes are automatic responses that are coordinated within the spinal cord. They are the basic building blocks of how our neurons function. They are rapid and automatic, and they have little variation in the response. So if a sensory information comes in, the motor response will go out, and it'll be about the same every time. Reflexes help to preserve homeostasis by making rapid adjustments in the function of organs and organ systems. They're interconnected through sensory neurons, motor neurons, and interneurons, and we have both simple and complex reflexes. So we already know that our sensory information gathers information in the peripheral nervous system and delivers it to the central nervous system, and that motor fibers are going to carry motor commands from the central nervous system to the peripheral nervous system. So one neural reflex produces one motor response. So this is called the reflex arc, which is the wiring of a single reflex. So it begins at a receptor. That receptor is going to send information to the central nervous system. the central nervous system is going to issue a motor command, and the motor command is going to end at a peripheral effector. So that effector could be a muscle fiber, like I touched a hot stove, I need to contract my muscles to pull my hand off that hot stove. Or it could be a gland saying we need to secrete something. In general, reflex arcs are negative feedback, so they oppose the original stimulus. So something happens and we want it to unhappen, so we're going to oppose that with the reflex arc. There are 5 steps in a neural reflex. So step 1, which you see up here, is the arrival of the stimulus. So in this stimulus, you have put your hand on a tack. So that's step 1. Step 2 is the activation of the sensory neuron. So that sensory neuron gets activated when a tack enters your hand. And so it's going to send information through the dorsal root ganglia to the posterior horn of the spinal cord. Step 3 is information processing within the central nervous system. So we have this interneuron shown in white, and the sensory neuron is going to synapse on the interneuron right here, and then the interneuron is going to go synapse on the motor neuron. So that's our processing, is the interneuron can do multiple things. So the first thing that it does is it synapses on the motor neuron, and the second thing is it crosses over and it ascends in the spinal cord and relays that information to the brain. But while this information is getting relayed to the brain, the motor neuron is already firing. So step 4 is that activation of the motor neuron, which is going to send a signal out to the effector. So in this case, the effector is the biceps muscle, and the effector is going to respond by contracting and lifting the hand up off of that tack. So while all this is happening, this information is going up to the brain, and eventually your brain is going to process that information as pain and say, wow, my hand hurts. But by the time you realize your hand hurts, you've already lifted your hand up off the tack because of this spinal cord mediated reflex here. Our reflexes can be classified in 4 different ways. So the first way is by development. Innate reflexes you are born with. They are genetically or developmentally determined. So you're born with that reflex, and or it develops very early on. So those would be considered innate reflexes. Acquired reflexes are learned reflexes. So I like to think about these as looking at a slalom skier. So a skier who does a slalom course, when you first go into a slalom course, if you're a new skier, you have to think about every twist and turn that you need to make to get through those slaloms to not fall. But the more and more and more you do it, you think less. You can start looking further ahead and understanding what your body has to do. And by the time you're an Olympic skier, all you have to do is keep your eyes on the prize, and your body knows exactly what to do, and you make all of those little reflex adjustments to get through those moguls without even having to think about it. And so that's an acquired or a learned reflex, is that ability to you do something over and over and over again until it becomes a reflex. We can also classify reflexes by response. So a somatic reflex is a control of skeletal muscle contraction. And our visceral reflex is a control of smooth muscle, cardiac muscle, or glandular tissue. We can classify by the complexity of the circuit, so how many neurons are involved. Monosynaptic is one synapse involving 2 neurons. Polysynaptic is several neurons, up to several hundred neurons that are involving multiple synapses. And then last, we can classify them by processing site. Spinal reflexes are processed within the spinal cord. Cranial reflexes are processed within the brain. So a specific reflex can fit into multiple categories. but we can look at them in multiple different ways. The complexity of reflexes is going to increase in order from simple, which would be monosynaptic, 2 neurons, 1 synapse, to polysynaptic, which is multiple neurons, multiple synapses, to the most complex intersegmental reflex arcs, which include many different spinal segments, and these intersegmental reflex arcs can produce a highly variable motor response. So monosynaptic, there's going to be no variability. It's going to be every time it happens, you have the exact same response. Polysynaptic, pretty similar. You're going to have about the same response every time. Intersegmental reflex arcs, the response are going to be much more variable because there are so many different moving parts to an intersegmental reflex arc. Stretch reflexes utilize 1 synapse, so they have the least delay between sensory input and motor output. So the patellar reflex is a stretch reflex. If you're sitting down and I take my reflex hammer and I tap on your patellar ligament, your leg is going to kick out and it's going to happen very quickly. So they're completed in about 20 to 40 milliseconds because the action potentials are traveling along those myelinated type A fibers, which are those super fast fibers. The receptor in the stretch reflex is the muscle spindle. So we're going to look at what a muscle spindle is. So muscle spindles look like this. They are bundles of intrafusal muscle fibers that are innervated by sensory and motor neurons. And they're surrounded by what are called extrafusal fibers, which maintain the tone and contract the muscle. So the intrafusal fibers are in the center of the muscle spindle, and they are wrapped around with dendrites of those sensory nerves, so that every time this muscle gets stretched, it's going to stimulate those sensory nerves to send information to the spinal cord. And then as soon as the spinal cord gets that information, they send information back in this gamma efferent motor neuron that says contract this muscle. And so that muscle is going to automatically contract. And this is a protective mechanism. When a muscle stretches, you're going to contract it so that you don't overstretch that muscle and injure it. So to look more closely at this reflex, the stimulus is the reflex hammer on the patellar ligament, which causes a stretch in the muscle spindle of the quadriceps muscle. So when that muscle spindle gets stretched, we are going to stimulate the dendrites of that sensory neuron that are surrounding the intrafusal muscle fibers. And that's going to send sensory information through the dorsal root ganglia into the posterior horn of the spinal cord. And that information goes directly to the anterior horn and synapses upon the lower motor neuron, on the gamma motor neuron. These are called gamma motor neurons there in the anterior horn. And then that gamma motor neuron is going to send information back to that muscle spindle that says contract. The muscle is going to contract and the foot is going to kick out. So we have the stimulus. It's going to stimulate the sensory neuron. The sensory neuron is going to synapse directly onto the motor neuron. The motor neuron is going to synapse back onto the effector and cause contraction, and the contraction of the quadriceps is going to kick your foot out. Gamma efferents are the axons of the motor neurons that leave the spinal cord, completing the reflex arc and synapsing back onto that muscle spindle to create contraction. Postural reflexes utilize stretch reflexes, and they help you to maintain an upright posture. When you're standing up straight, like say I was standing in front of the class lecturing right now, I don't have to spend any of my time thinking about standing upright. You can just stand upright and not think about it. And that is because we have these these stretch reflexes within our muscles that as soon as a muscle starts to stretch, like if you start to sway backwards, and then you're going to feel that in the muscles in your anterior body, so they're going to reflexively contract to bring you back upright. And if you start to sway forward, the muscles in the backside of your body are going to contract to bring you back to neutral. So they're automatically maintaining your balance. Polysynaptic reflexes utilize more than one synapse, so they're more complicated than that monosynaptic reflex. They involve interneurons, and they often control more than one muscle group. They produce either excitatory postsynaptic potentials or inhibitory postsynaptic potentials, which means they can either facilitate something that's happening through excitatory postsynaptic potentials, or they can inhibit it. The tendon reflex prevents our skeletal muscles from developing too much tension and therefore tearing or breaking our tendons. Sensory receptors are distinct from muscle spindles or proprioceptors. They, when they are stimulated, they're stimulated when collagen fibers begin to overstretch. That is going to activate inhibitory interneurons in the spinal cord. Those interneurons control the skeletal muscle, preventing it from developing enough tension to break its own tendon. So when the collagen fibers of the tendon start to stretch, they send a message to the spinal cord that says you need to relax this muscle so that we don't break. We find these in weightlifters or big bodybuilders. If you've ever seen these guys who are trying to lift like say 400 pounds or 500 pounds, they're working, and then all of a sudden their muscles are just going to give out and they're going to drop that weight. And that's because their tendon reflex kicked in and was a protective mechanism to keep them from snapping their tendons. The receptor in the tendon reflex is called the Golgi tendon organ. So it's Golgi like Golgi apparatus, but Golgi tendon organ is the receptor in the tendon reflex. The withdrawal reflex is where you are moving a part of the body away from a painful stimulus. So this is what we looked at earlier with putting your hand on a tack. So this is called the flexor reflex, where you're pulling your hand away from a hot stove. This is also the same if you're pulling your hand off of a tack, if you were to lean on a tack. So the strength and extent of the response is going to vary depending on the intensity and the location of the stimulus. So we'll go over this again, that here you've grabbed onto this hot pan, and it stimulates your sensory nerve through the dorsal root ganglion to the posterior horn of the spinal column. And there it synapses on an interneuron, and that's what makes this polysynaptic. On the monosynaptic, that sensory neuron went all the way to the ventral horn and synapsed directly onto the gamma motor neuron. But in this instance, we are synapsing on an interneuron, and then the interneuron is going to synapse on the motor neuron, causing flexion, but it's also going to send information superiorly up to the brain to so that you can perceive the pain that happened when you burned yourself on that pan. Now we're going to get a little bit more complex. So in order for the flexor reflex to work and for you to be able to flex that bicep and quickly pull your hand back, you need to inhibit the antagonist muscle, which in this case would be a triceps muscle. So in order for the flexor to work, you have to inhibit the extensor. And this happens through a process called reciprocal inhibition, and it's performed by the interneurons within the spinal cord. Ipsilateral reflex arcs occur on one side of the body. So the stimulus is on one side, the response is on one side. And then we have the crossed extensor reflex, which is our most complicated reflex, and that involves a contralateral reflex arc where you're utilizing both sides of the spinal cord, both sides of the body. So the cross-extensor reflex occurs simultaneously with the flexor reflex. So the flexor reflex is ipsilateral, then the cross-extensor reflex is a response on the contralateral side of the body. So this one is showing us stepping on a tack. When you step on a tack, it is going to stimulate your sensory neuron, and the sensory neuron is going to come and synapse on an interneuron in the spinal cord. We've seen this before with grabbing onto a hot stove, so the flexor reflex is going to occur on the right side of the body because you stepped on the tack with the right foot. So on the right side of the body, you've synapsed in the interneuron, and the interneuron has synapsed on this lower motor neuron, gamma motor neuron, and the gamma motor neuron is going to stimulate the flexors in the leg to pull the foot up off the leg, off the tack. Let me say that again. You stepped on a tack. The sensory nerve is going to synapse on an interneuron, and on the ipsilateral side, the interneuron is going to stimulate the flexors to pull the leg up off of the tack. And then it's also going to inhibit, in blue here, it's going to inhibit the extensors so that you're not fighting your flexors with your extensors. So there's two things happening on the right side of the body. You are stimulating or giving an excitatory postsynaptic potential to the flexors to lift your leg, and an inhibitory postsynaptic potential to the extensors to allow for quick flexion. So that is the flexor reflex on the right side of the body. Then on the left side of the body is the crossed extensor reflex. So the same interneuron crosses over to the left side of the body and synapses on two neurons in the anterior horn. One of them is an excitatory postsynaptic potential to the extensors in the left leg to stimulate those extensors to contract so that your left leg can hold the weight of your body when you lift up your right leg. The other one is an inhibitory interneuron, which is going to inhibit the flexors on the right side, again allowing for that quick and sustained extension of that left leg. So the legs are more complicated than the arms, because in the arms you just have to pull one arm away. But in the legs, if you flex one leg, you need to extend the other leg or else you're going to fall over. So we have the flexor reflex, which is stimulating the flexors on the right side and inhibiting the extensors on the right side. And then the crossed extensor reflex, which is stimulating the extensors on the left side and inhibiting the flexors on the left side. Okay, we have 5 characteristics of polysynaptic reflexes. So polysynaptic, more than one synapse. They involve pools of neurons. They are intersegmental in distribution. They involve some type of reciprocal inhibition. They have reverberating circuits, which prolong the reflexive motor response. So in our crossed extensor reflex, the reverberating circuit is going to allow us to continue to stand on our left leg as long as the right leg is up in the air. And there are several reflexes that cooperate to produce a coordinated and controlled response. Now, these reflexes occur in our body in response to a stimuli, but the brain can also alter the spinal reflexes, which is really cool. And the brain can either facilitate them or utilize them for its purposes, or it can inhibit them if it doesn't want them to happen at that time. Higher centers of the brain incorporate lower reflexive motor patterns. So automatic reflexes can be activated as the brain needs, and can use fewer nerve impulses to control more complex motor functions. So walking, running, and jumping, when we're doing these things, we're utilizing our normal reflexes to make a more complex behavior. Higher centers can reinforce spinal reflexes through facilitating or inhibiting those motor patterns in the spinal cord. So one thing that I think is so cool, and to show you how this works, is consider somebody walking on a tightrope between two skyscrapers in Manhattan. Picture that person being you. So you're in Manhattan, maybe not right now, but in general, we'll say it's healthy time and you're in Manhattan and you walk across this tightrope and everything's going well, and then a bee comes and stings you. Your reflex when a bee stings you would be to swat at that bee and rub your arm. But if you're on a tightrope and you do that, you're likely to fall and plummet to your death. So your brain can override your reflexive desire to smack that bee, and it can let that bee sting you in order to preserve life and keep you on that tightrope. So that's how your brain can suppress or inhibit some of our normal reflexes if it's not in your body's best interest at that time. A single excitatory postsynaptic potential may not depolarize the postsynaptic neuron sufficiently to generate an action potential. However, it makes that neuron more sensitive to excitatory stimuli. This is called facilitation. So a single excitatory postsynaptic potential can bring that neuron closer to its threshold potential so that another smaller depolarization can create that action potential. So that is called facilitation. Reinforcement is the enhancement of a spinal reflex, and inhibition would be the inhibition of a reflex like we just talked about in the last slide. The Babinski reflex is normal in infants. So in an infant, if you were to, and this is super fun to play with, so if you have a baby that you have access to, I would encourage you to go mess with it. I know that's mean, but I like messing with babies. So you can just use your finger. You don't have to use a reflex hammer. But if you use your finger and you swipe from the heel towards toes in the bottom of an infant's foot, they're going to splay out their toes. Whereas if you do it in a normal adult, they're going to clamp down their toes and a protective mechanism. If you have an adult who's had an injury to their central nervous system, so a brain injury or a spinal cord injury that could be above the level of the legs, so that could basically be any spinal cord injury, they are going to exhibit this Babinski sign, which means when you swipe their foot as an adult, they're going to splay out their toes instead of clamping their toes down. So this is another way that we can look at is, so say somebody's having pain down their leg and we think that it's a disc herniation, maybe we think that that herniation is pressing on a spinal nerve, in which case the plantar reflex would be negative. Or if it's pressing on the spinal cord and causing problems with the actual spinal cord, then their Babinski sign will be present, and that will indicate a lesion of the central nervous system. Okay, that ends our lecture on the spinal cord, spinal nerves, and spinal reflexes. There's going to be an extra assignment this week. Be sure to look for that, which is going to be writing out the steps of the cross extensor reflex. I want to make sure that everybody understands exactly how that happens. So it'll be a step-by-step process of that, and that will be your lab grade for this week. And then next week we are moving on to the brain. So we're getting excited. 2 more lectures to go. I know you guys can do it, and I will look forward to seeing you next week. Have a great week.